developing molecular phylogeny using subgen sequencing data for elucidating some topics concerning its systematics and evolution. So it took this title for the talk, Systematics of Prosthechea, reassessing the lumpers, splitter debate, and shedding light on the evolution of this neotropical lineage through a molecular phylogeny. So it should be working, this thing. Perhaps. Okay. Sorry. So here's a presentation, the presentation summary of this talk. In the first one, we'll be talking about the aspects of the diversity, distribution, morphology, and ecology of this orchid group. Then I'm going to get into and bring you guys into the taxonomic history of this genus and this lumper splitter debate concerning its circumscription. To then get into the results that it could achieve with this molecular phylogeny. So to split or not to split Prosthechea. And then I'm going to get into some biogeographic considerations. And at the end, I'm going to talk really briefly about what I'm focusing on during this H2H fellowship here at Harvard. So Prosthechea is a neotropical orchid genus belonging to the subtribe Laeliinae, which is a highly diverse subtribe within the Orchidaceae family, also exclusively neotropical distribution. And currently, there are about like 120 species of Prosthechea described so far. The genus has a neotropical distribution, as I mentioned. And the plants can occur in a wide range of types of habitats, from sea level to about like 3,000 meters of elevation. But I would say that most of the diversity, most of the species of Prosthechea, they occur in mountain forest habitats, especially the cloud forests. So there's three main diversity centers for the genus: Mesoamerica, the tropical Andes, and the Brazilian Atlantic forest in eastern Brazil. So here are some pictures highlighting all this diversity in terms of floral morphology. And yeah, so in terms of life forms, Prosthechea predominantly has epiphytic plants, so plants that grow on other trees, but some species are also adapted to growing on rocks as lithophilic life form, and a few other species also as terrestrial, mostly in those kind of roadside banks. It's kind of with some sort of declivose situation. About the
how many terrestrial species?
Sorry?
How many terrestrial? How many?
Maybe, I don't know numbers, but let's say like 20 species. But there's also some species that are epiphytic, and eventually they are facultative terrestrial also. So about the morphology of the plants in terms of the vegetative aspect of that, they can show like a cespitous growth, where the symphodia are clustered in a way that you can like really figure out the rhizome. And there is also plants of more rhizomatous growth, where you can like clearly distinct the the pattern of the rhizome, and then the susceptible symphodia growing from that. The stems are swollen in pseudobulbs, these structures of reserve, common in orchids. And they can vary a lot in terms of shape from like short and ovoid to long and slender. And it's quite common that they usually present some degree of lateral compression. This is a very peculiar trait of the genus. About the inflorescence, predominantly simple racemes, and eventually some species can present like compound racemes, like panicles. And nowadays, the inflorescence is subtended by a spathaceous branch or spathe. They usually develop first, and then after that, the inflorescence emerges and develops. About the floral morphology, it's quite variable, and the flowers can be resupinate, as in this lower row. So in orchids, we have this phenomena of resupination or non-resupination, where it pretty much says about the final position of the labellum or lip in anthesis. So there's species with resupinate flowers where the lip is in lower position at the display moment, whereas other species in the genus present non-resupinate flowers, so the lip is in the uppermost position at the moment of the display. Get into more like details of this floral morphology. This is like synapomorphic to the genus is the presence of three-tooth lineage. So the tip of the column is there is also these three, projections. It's called the tip of the column. So kind of like nails, like holding the antler. So it's even among all these variation in shapes and forms, they always preserve these three-toothed lineage. And the mid-tooth usually is fleshy, appears like a fleshy appendage. So this swollen part of the mid-tooth. The callosity of the labellum is usually simple. So as in this example of Prosthechea bonkiana, just as a sort of, it's something that we call a padlock, just like an elevation, can be pubescent or glabrous. And the number of pollinia are 4. So we have also some variation in terms of the callus morphology, can be more complex. But overall, most of these species present like a more simple callus. Another remarkable trait in the genus is the morphology of the fruits that are usually three-winged or at least strongly three-angled. So there's these hues on the fruits and this very peculiar morphology. And the suture zone between the carpels, there's this strap of tissue that covers this part, and usually it's from where like when the fruit opens, from where the seeds are released. So here, different species that present this, common trait. Now starting to talk a little bit about the taxonomic history of the genus. The type species is Prosthechea glauca. It's a Mexican species described at the beginning of the 1838. And at that moment, it was pretty much some segregations of Epidendrum, which is another orchid genus, like the Megalibus orchid genus. So at that point, some plants that usually were traditionally recognized as Epidendrum, they were starting to be like described as new genera. So Knowles and Westcott described like Prosthechea glauca, proposing this new generic concept. And the etymology of the genus, it's about this, as I mentioned, this mid-tooth appendage, like swollen appendage, that in Greek, prosthech means appendage. So putting in a timeline this taxonomic history. So in 1838, Knowles and Westcott described this Mexican plant, proposing like a new genus, Prosthechea. 2 years later, Lindley, which was like a very prestigious and influent orchidologist of the United Kingdom, he was like, if I'm not wrong, director of Euchile for a while. He combines under epidemic, which as a commenter had like a really broad circumscription at the time of epidemic. So 2 years later, it was just treated as a synonym of epidemic, Prosthechea, this concept of genus. A little bit later, Lindley published some infrageneric classification for Epidendrum, subdividing the genus in 10 subgenera. And another proposition of splitting a group of Epidendrum was proposed by Reinhold, like segregating Hormidium. The point here is that most of the plants that I'm going to present in this timeline are currently circumscribed as Prosthechea. I'm going to get there. But just to illustrate how Epidendrum has like a really broad sense at that point of the taxonomic history, and that afterwards several other authors tried to split groups from Epidendrum, but always with Lindley after all putting in the synonym of that. So a year later, in 1842, Hoffmann proposed like Anacheilium to segregate also a peculiar and weird epidendrum, let's say. And in 1853, there is like a really important taxonomic treatment of Lindley to epidendrum, where he subdivided epidendrum in several subgenera. And he combined all these names again under epidendrum. And here, just to illustrate the current circumscription of Prosthechea, we could find species into these 5 subgenera of Lindley. In 1914, Schlechter reconsiders for the first time another name, again trying to split things from Epidendrum and the genus Encyclia. In 1936, Oak Ames do not recognize this segregation of Encyclia from Epidendrum, and his the genus Epidendrum in the United States and Middle America still recognizing that broader circumscription of Lindley to the genus. And in 1960, Brigger recognized Hormidium in again as trying to put a part of Epidendrum. And just in 1961, Robert Dressler proposed like the first and more comprehensive reconsideration of Encyclia, trying to split this broad sense of Epidendrum. So he subdivided Epidendrum and Encyclia based on column, morphology traits, and also by the fact that usually in Epidendrum, the claw of the leaf, the nipples of the leaf is completely fused to the column, whereas in Encyclia, it's just partially. And in a sectional classification of Dressler, he subdivided Encyclia in 2 sections. So section Encyclia and section Osmophytum, based on morphology of the crenundum, so like 3-tooth crenundum with deep sinuses for Encyclia section Osmophytum, and a crenundum with not so conspicuous teeth for Encyclia section Encyclia. In addition to the morphology of the fruits, they are strongly twin or triangle in Osmophytum, and cylindric in secret section in secret. Afterwards, they just elevated this subgenera, this section to subgenera, and then in 1981, Pabst and colleagues, he was like a Brazilian orchidologist, tried to propose again the reconsideration of Anacheilium, trying to circumscribe those species treated by Dressler into Encyclia subgenus Osmophytum. But Pabst did just a few of name combinations, so lacking a bunch of names behind. And just in 1997, Higgins clarified that the oldest name related to the Encyclia subgenus Osmophytum is actually that old name Prosthechea glauca from the beginning of the 19th century. So then Higgins proposes reconsideration of Encyclia, and pretty much what Dressler was considering as Encyclia subgenus Osmophytum is now treated as Prosthechea, which is the current circumscription of the genus, and what I'm going to discuss about. So at the turn of the 21st century, Prosthechea was reconsidered and circumscribed, as I mentioned, and it starts to be sampled in molecular phylogenies at the level of subtribe and family, but always just including a few species. If you think that the diversity of the genus is about like 120 species. So in the more recent of those, in 2009, Prosthechea was reported as monophyletic group. In all those other phylogenies, it always was reported as monophyletic group. And in this phylogeny, in this clade, that was called Encyclia alliance. So Napolitani together with Encyclia and Alemannia and Anacheilium, other related genera. But after that, some alternative classifications were proposed. Pretty much these two classifications, one in 2003 and another in 2004. Pretty much trying to make use of these older proposed segregations, like Anacheilium, like Hormidium, and also proposing new names to split this broad sense of Prosthechea into a thinner classification. So Piron and Cassonet in 2003 pretty much divided Prosthechea in two genera. So Prosthechea in a narrow sense and Pseudencyclia, and also Euchile that were already proposed before, but they even consider in the discussion of the work. And Wither and Harding proposed a subdivision of Prosthechea as circumscribed by Higgins into 6 genera. So using several morphological traits, all these classifications were proposed pretty much based on morphology exclusively, not considered like any phylogenetic aspect. And then some questions were kind of open. So like, would Prosthechea as circumscribed by Higgins still be recovered as a monophyletic group after a global sampling? Like, do these smaller proposed genera such as Anacheilium, Euchile, Hormidium, Panarica, Pollardia, and Prosthechea in a narrow sense, would these smaller proposed genera reflect monophyletic groups in a phylogenetic and a more like robust phylogenetic analysis? And what about the evolution of remarkable morphological traits within the genus, such as the resupination or the non-resupination of flowers? So to address these questions, during my PhD, we developed this phylogeny to the genus. So this is the title of the work, Phylogeny of Prosthechea, based on ITS and plastic DNA sequences, reassessing the local speed debate, and shall let it into this Neotropical genus evolution. Coincidentally, it was just published like this last weekend. It comes as an early version from Taxon. So check it out. And so for sampling, we could get 80 species of Prosthechea, which is pretty much like 3/4 of the genus diversity, and 11 species of related genera, like including the species previously sampled on the Encyclia alliance, and also related species present in other topologies of phylogenies dealing at the subtribe level. And we took Apophyllum giganteum as outgroup for the analysis, since it's the sister group of all other remain Laeliinae. So it's the early lineage within the subtribe. So summarizing 92 terminals, OTUs for this analysis. So to reach such sampling, I went to several living collections in Brazil, such as the orchid living collection of the Instituto de Botânica in São Paulo, a very important collection. Also in the Exalc USP collection in Piracicaba, São Paulo, where Vieira was working during his time, and there's a bunch of plants of Prosthechea and cultivation there. Botanical Garden of Manaus in the Amazonas, and also the Rio de Janeiro Botanical Garden. Additionally, I visited some collections also in Mexico, since there's like Mesoamerica is also a center of university, so a bunch of material that I could like collect in Brazil, I did in Mexico, in important living collections such as the Mexican Orchidology Association and the living collection of the Autonomous University in Mexico City. And also, I did some field work to collect plants. So there was some field works in the northeast of Minas Gerais in Brazil, where there's some like pre-endemic species, such as Prosthechea tabeniensis, that is known for just like one isobed over there. Prosthechea paxepala in the region of the Padre Angelo mountain complex. There are species epiphytic in Abelosia. Also, some field work in the Atlantic forest in southern Bahia. So, sites Serra da Pedra Lascada in Barro Preto, where you could collect these southern Bahia endemic Prosthechea suluana. Other sites there in Bahia, such as Serra da Boia, National Park of Una, where there's some areas with mountain forest formations. Also, the Reserva Bonita, the Serra Bonita Private Reserve in Camacan, real realm of diversity in southern Bahia. We collect a bunch of material over there. Also, some endemic species such as Prosthechea bankiana, Prosthechea borealensis, Prosthechea silvana. And also in Chapada Diamantina in Bahia, more to the countryside of the state, where we have these the campus rupestris, these portizitic formations where there's some plants adapted to these litophytic life forms. So you can collect also some endemic species of the region, such as Prosthechea carri, Prosthechea mugeni. And then to the bench work. So most of the bench work was done at the State University of Feira de Santana, the Lamol lab. And part of the lab work was done also in Mexico. So it was pandemics. And I was planning to go to Mexico. It was kind of February of 2020, just when everything turns upside down. And I was really needing to get those samples from Mexico. My collaborator over there, Professor Gerardo Salazar, had a bunch of material in his DNA bank also. So that was kind of postponing during the pandemics. And when then things become a bit more like relaxed, let's say, in late 2020, I went to Mexico City. The Autonomous University was closed, but Gerardo pretty much set up like a lab at his place. So I was living at his place and doing bench work at his home for 2 months, working with the samples. And he got one of these mini PCR machines. It's like a small thermocycle that you can put like at your hand. The same one that people use on the spatial station. So it has like 16, holes. So it's pretty much in a bunch of PCR 16 per 16 samples. And after all, we could like get to the sequences. So the combined data set, like including, and the markers that we worked was ITS, RPL, 32 turn L, turn D, turn T, and the 3' portion of the YCF1 subgen plus placebo subgen. So the combined data set was something about like 5,000 base pairs. And we did analysis using Bayesian inference and also maximum parsimony. We were like interested in maximum parsimony to be able to perform this partition Bremer support, which is a kind of analysis of the K index where you can kind of measure at each node of your tree how much weight of a certain partition influences on that topology recovery. Like looking for investigate potential, conflicts between ITS and Protestant partitions. So we relied our description, interpretation of the results on the variation tree of the combined data set, just taking account nodes with procedure probability above 0.9 at least. So this is like the topology that we could look over, where Prosthechea was recovered as a monophyletic group with strong support of posterior probability and kind of weak support of bootstrap analysis of the maximum parsimony analysis. We could not recover exactly what is the sister group of Prosthechea, since the genus is in this polytomy together with a clade including Laeliinae and Amoana, another clade including part of Westlumbia and Encyclia, and Westlumbia luteorosa as a divergent lineage here in this polytomy. So getting into the description of the circumscription of the genus. So when we map all those alternative classifications in the phylogeny, we can check that, first of all, the classification of Chironia castromata subdividing Prosthechea into two genera pretty much. It's both proposed genera are polyphyletic, as you can see here. And when you get to the classification of Leaeliinae Harding, that subdivided Prosthechea into 6 genera. 3 of those genera were recovered as monophyletic groups, which are Euchile, Hormidium, and Panarica, with the detail that all these genera represent really small plates with a few species, not more than 10 species. For like Euchile, it's pretty much all just these 3 species for the clade. Of Hormidium, maybe there's other 1 or 2 species that students sample that likely would fit there. And the same for Panarica, which is lineage concentrated in Mesoamerica and the name is derived from this so it's like Panarica from Panama and Costa Rica, but the plants also reach in Mexico. But the thing here is that it's like small plates with like homogeneous like morphology. Whereas the other proposed genera are polyphyletic, such as Pollardia, as you can see here, polyphyletic. There are narrow stems of Prosthechea, also polyphyletic, and also Anacheilium, polyphyletic. There's a detail that there's like a polytomy at this node C of the tree. So it kind of limited a bit how deep we could go into in terms of analysis. And also, given this configuration, one would what about if these clades E and F with more data are recovered together with this bigger clade M? Even though Anacheilium would be like paraphyletic, since Hormidium would be nested within that. And the interesting here is that between Anacheilium and Hormidium has a priority because it's an overload. So in the future, with more data, if it's the case of segregating Prosthechea and the more genera, this idea of this big group, this potential big group, if in the case of E, F, and M being recovered together, the most appropriate name would be actually Hormidium. However, what to do with these, other clades? So just to illustrate, this clade I, present plants, in addition of having like Panarica in the middle of the thing, has plants with entire leaves, three-lobed leaves, non-resupinated flowers, resupinated flowers. So a wide range of variation in just like a single plate, which kind of reflects how complex it is to fit, this group into, even if you want to put that in, a bunch of smaller blocks, it will still be kind of complex based on morphology. And also, we didn't see like advantage in proposing even more names to a group where there's already like a bunch of associated names. So we basically defend a broader sense of Prosthechea as originally proposed by Wesley Higgins. And instead of, splitting the genus in a bunch of small groups. So we did also some reconstruction of morphological traits in the phylogeny, such as the resupination. So non-resupination evolved like more than once. There's this big, clade with non-resupinated flowers within the genus, but it also evolved like independently in another situations during the evolution of the genus. The same for leaf lobulation. We also try to, so we are pretty much like trying to track, morphological traits that were used to, to propose this final genetic treatments, how they would be like reflected after, like ancestral stage reconstruction. So for leaf lobulation, the same, you have like a deep plate with entire leaves, but also entire leaves is moving independently several times during the evolution of the group. The same for type of callus. We have a big clade with simple kind of callus, but it also evolved independently on other times. And the same for inflorescence. And for the only trait that I could like recognize as some like phylogenetic pattern was the inflorescence architecture, because the most common pattern in the genus is like the simple racemes, and just once it seems to have evolved with like compound racemes, panicles, even though there's some polymorphism, so kind of complex in terms of morphology. When you look to the biogeography, let's say how distributed are the species of the clades, like the earliest divergent lineage within the genus, the clade A is endemic to Mexico. Clade B occurs in the West Indies and in the Yucatan Peninsula, also reaching Prosthechea bucciana, also reaches southern Florida. Actually, it's one of the few species of Prosthechea that reach the US. So Prosthechea bucciana and Prosthechea Pollardia also reach southern Florida. So the clade B essentially of Yucatan Peninsula distribution and West Indies reaching Florida. How these clades highlighted in yellow, they are essentially of Mesoamerican distribution, with a few species reaching South America or West Indies or other areas. The clades P and R are are distributed across Mesoamerica and South America, also reaching other areas such as West Indies. And here, this clade S is essentially of South American distribution, whereas the clade V is endemic to Eastern Brazil. So we see this pattern of early divergent lineages within the genus of more Mexican or Mesoamerican distribution, more recent lineages endemic to Eastern Brazil. So this is not this isn't published data. This is not on the article that was just published, but it's some data that I presented in my thesis. So we perform like a reconstruction of ancestral geographic distribution using this Bayesian binary MCMC analysis. It's pretty much the only analysis where you can perform reconstructions, even having like a polytomy in the tree. So we subdivided the Neotropical region in 9 bioregions. And pretty much what we retrieve here is that Prosthechea has probably a Mexican origin. And here we subdivided, on the Tehuantepec-Isthmus is bioregion 8, which is Mexico, and like Central America extending to Yucatan Peninsula. So there's likely a Mexican origin for Prosthechea. And afterwards, the genus was dispersing across the Neotropical region towards southern Brazil until reach like the Atlantic forest here. So this is an interesting result since it kind of puts Mesoamerica as a cradle of diversity, which is a pattern that contrasts with this idea of the Amazon as always being the main source of diversity to the Neotropical floral assemblage, which was presented in the paper of Antonelli and colleagues in 2018. So Prosthechea and other lineages in Laeliinae and also in the subtribe epidemic in Orchidaceae show this kind of pattern of having a center of origin in the Mesoamerican region and afterwards dispersing to the South America. This is also published data, some analysis that I have been working more recently, still exploring this data of my PhD. So this is a maximum credibility clade credibility tree generated beast with 2 secondary calibrations. So it's estimated that the origin of Prosthechea took place about 8.14 billion years ago during the late Miocene. And we also can notice a bunch of diversification events during the Pliocene and also during the Pleistocene. And so here, as a HUH fellow, I have been pretty much like focusing on improving this phylogeny, now working with phylogenomics. So during the last year, I have been pretty much like generating this data. We are using the Orchidaceae 963 Bates kit, which was published in 2021, that provides hundreds of nuclear orthologous genes. So we'll be working in this hybrid approach, like mixing the target information with genome scheming. I just got data. Part of the data I took in mid-December, and the other part was like in some the multiplexing by the bioinformaticians from Bauer. They just gave to me recently, maybe I think last week, if I'm not wrong. So from now on, I'll be pretty much like working on developing this and on analyzing and putting together, the products of this data in terms of publications. And also I have been really interested in the systematics and taxonomy of a species complex distributed through the Andes, Central America, and West Indies. So I could put a bunch of samples using herbarium materials here at Harvard and other institutions here in the US into these sampling for these phylogenomics. And yeah, so pretty much from now on, I'll be working on analyzing this data and, figuring out what sort of products can be present from that. So I would like to acknowledge the State University of Feira de Santana and its grad school in botany, where I developed my PhD thesis. I also would like to acknowledge CAFES for my PhD scholarship and CNPq for the grant that funded most of the molecular work that I present to you guys so far of this published paper. Also to Fundação Flora de Apoio Botânica, of what I was a grantee during 2006, from 2016 to 2017, where I spent like a year on the National Museum of Natural History, working on the digitization of the Brazilian specimens over there. So the first 6 months of my PhD, I was there, and that was the beginning of my, how could I say, international network, Also to Harvard University, that at that point granted me a travel grant to visit here in the collection in 2017. To Cássio Vandenberg, my former advisor, PhD advisor, and Gerardo Salazar, collaborator of this project. And here names of the people that facilitated or provided samples of Prosthechea to the development of this work of this phylogeny. So thank you for your attention. I hope you have enjoyed this presentation.
Questions for Tiago? Tiago, you can repeat the question so people online can hear.
Repeat like.
If someone asks.
Oh, okay. So yes.
I have two related questions. So I thought it was fascinating to see so much morphological diversity than image right now in the floral morphology, but also the morphology of the pseudobulbs and the life forms and where they can grow, right? And I was wondering if you have plotted like those traits in your new phylogeny to see if there are some correlations between traits, like between the vegetative morphological ones and the floral ones, which leads me to the second question that I have, and is like, what do you think is behind so much morphological variation? Is it that like in your resupination phylogeny you have like one group here and then one other one there, then a big one? Like, do you think is it like selective pressures are making these plants diverge in morphologies, or is it more of a random process that like a weak selection that allows these plants to vary so widely?
So the first question was about pretty much using vegetative traits in reconstructions. Okay. And the second question was about what would be driving such a diversity and kind of random pattern of floral morphology throughout the phylogeny. And vegetative, okay. So about the vegetative traits, it's kind of tricky because they can be pretty polymorphic, sometimes. So yeah, I tend to reconstruct more vegetative traits, but I would say that it's for sometimes it's kind of tough because not all species I have contact in the field to see how they naturally grow. Sometimes I just have like a balance sheet where it's certainly kind of suffer in terms of its morphology. But yeah, it's certainly I'm really looking forward to have these results of these phylogenomics to see if what about like the topology, if things will change a lot, what's going to happen, and about the what would be driving such a pattern of of types of morphology spread across the phylogeny. I think that will be related to the ecology of the group in terms of pollinators pressure. So sounds pretty much like some sorts of like pollination syndromes that different lineages had to get adapted to be able to still have those pollinators working with But something that I also think that it's, and after all, I forgot to mention, is that a potential scenario of reticulate evolution in the genus. So I didn't mention, but the positional grammar support analysis, despite they're not like conclusive in terms of reticulation as the reason of certain conflicts between partitions. I mean, it could be just like incomplete lineage sorting of the velocity. But I don't know, based on what I see and the fact of orchids, they tend to be easygoing in terms of hybridization. I have a strong feeling that past civilization events have like a key role on the evolution of the group. And for the same reason, sometimes the morphology becomes kind of a bit chaotic. So it's even more complex to fit this group in easily recognizable morphological synaptopathies. Yeah. So Kanchi.
More or less all of the statements. One is the wind flow in orchids, whether the fruit is big or not, it's not going to get impact. This first of the flow. So how does that wind nature is in any way helpful or even a hindrance in the designs? If the fruit has to split open, whether the wing is in any way helping or not helping. Right. And regarding resupination, so you mentioned that some resupinate and some do not. Which one is evolved, and any observation on the pollination biology? The insects need to be different.
Yeah, I mean, there's very few studies dealing with pollination of Prosthechea. Just some like really general works. There's some works in southern Florida where it produce like this Pollardia, Euchile. And overall, what is published so far describes really without precision, just like some Hymenoptera, bees, wasps pollinating. So it still needs more studies concerning the pollination systems within the genus. It's a completely open field of work. But yeah, likely there's some reason, right, for this resupination or non-resupination, but it's still something that needs to be studied.
Biogeographic analysis. The results indicate that the genus has originated in Mexico.
Exactly.
And then the follow analysis that it did 2023, it was about 8 million years ago. And even that orchids have like a capsule and the seeds are very small, how would you hypothesize that this genus came from Mexico to like eastern Brazil?
Likely through Anacheilium's like dispersion. So sounds like originally the lineage was kind of a lineage adapted to more mountain sort of environments. So it was dispersing from mountains, Mexican mountains, the Mexicans of Central America, then to the Andes, and then spreading to reach like eastern Brazil in a successive system of dispersal through the wind. But this is the idea. I still need to explore and work on this sort of analysis and to reach more like robust conclusions about it. But talking about orchids, it's pretty much dispersing through the wind. Yes, so
To cross a resupinate plant with non-resupinate plant. What is the offspring of that?
that's a good question. As I'm not sure if someone did this sort of experiment. But yeah.
It seems like an easy. A lot of people cross references.
Yeah.
Somebody might have done that.
Maybe, I don't know. Maybe it should predominate like the morphology of the mother plant, that receives the pollen. But yeah, I really don't know.
No, it's just, if you're talking about hybridization, that's what happens.
In this case,
Well, yeah,
well, I would have to do the.
somebody would have.
Yeah.
I wasn't expecting you've done it, but for somebody.
Yeah.
Somebody had.
Yeah, something to be done.
Since you are using the sense of census dogs, it's a suggestion. Instead of abbreviating the second letter, just put one letter, use STR.
Yes. I see, yeah.
Because we usually abbreviate a term in front of a rover, not in front of a person. So, census, yes, STR.
Yes, yeah. Okay. So, any further questions?
Thank you, Tiago.